So in section 15.4, we're looking at step 3, the antigenic challenge of T cells and B cells. And so in this process, in this section, we are going to look at what happens when the presentation of the antigen occurs and they activate the B cells and the T cells, and what kind of cells are produced by the B cells and what kind of cells are produced by the T cells. So we're going to look at both the CD4 and the CD8 cells, and then we're going to look at the B cells. So these are the different cells that are secreted when the CD4 cells are activated. These are the subsets. And the memory cells are going to replace any CD4 cells. They don't replace CD8 cells, but they will replace any of these cells that have the CD4 marker. And so specifically, like the follicular helper is going to activate B cells. So it's going to secrete cytokines and things that will cause the B cells to become activated. So this is the T cell dependent, or T dependent method of activating B cells. So T dependent method of activating the B cells. And you can see these are the 3 types of cells that B cells will produce when they are activated. So they make regulatory B cells, which actually will suppress the T cell response, and plasma cells and memory cells. So depending on what is needed, this is what they're going to make. So memory cells, B memory will replace B cells. Plasma cells will make antibodies, and regulatory cells will actually suppress or may suppress the T cell response if needed. So T helper 1, they are known for activating the cellular immunity. So this T cell is the key, T helper 1. It's going to activate all the other T cells, the cell-mediated immunity. It's also going to activate macrophages, and that will make them more inflammatory. That's like makes them angry macrophages, is the way that I have always learned it and understood it. So they secrete cytokines that kind of activate the macrophages, stimulates them to become more inflammatory. This is also involved in the delayed hypersensitivity reaction, and so we see these T helper 1s being involved in that. Now, T helper 2, they don't really show what goes on here, and this is not really as much. It can be involved in delayed hypersensitivity, but really the T helper 2 is involved in the responding to extracellular microbes, allergens, and eukaryotic organisms, so protozoans and parasitic worms, so parasites, eukaryotic parasites. So that is really what they're known for. And eosinophils, increasing their amounts, have to do with the allergens and the extracellular eukaryotic microbes. So all of that is what we understand now about the T helper 2. So some of my previous notes and understanding of what goes on here with T helper 2 is a little bit changed because this has been redefined in the last few years. T helper 17 still is known for inflammation because it produces interleukin 17. And interleukin 17 causes the inflammation. And then T regulatory, is going to dampen the immune response as appropriate. So if there are still a lot of antigens present, then they are not going to dampen it. But if there are not a lot of antigens or bacteria or viruses, microbes, whatever, present, then there's no need to make all these other T cells. And so it dampens the immune response when we don't see a lot of microbes present. It also has this dual function of preventing us from attacking our own normal microbiota. So we want that normal microbiota to be present. And it's amazing that with all this immune system cells that it doesn't attack the normal microbiota. So those are a little bit about the T cell events. And here's an animation on T cell activation and the response.
Activation of the immune response typically begins when a pathogen enters the body. Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces. Macrophages with antigen fragments displayed on their surfaces are called antigen-presenting cells. An antigen-presenting macrophage interacts with a T helper cell that can recognize the same antigen. During the interaction, the macrophage releases a chemical alarm signal called interleukin-1, which stimulates the T helper cell to secrete interleukin-2. Interleukin-2 causes the proliferation of certain cytotoxic T cells and B cells. The immune response from this point follows two paths, one using cytotoxic T cells and one using B cells. Normal cells of the body that become infected can also digest some of the pathogens and display antigen fragments on their cell surfaces. The body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular antigen. The cytotoxic T cells that are capable of recognizing the antigen displayed on the surfaces of infected cells bind to the infected cells and produce chemicals that kill the infected cell. Death of the infected cells results in destruction of the pathogen. B cells also come in millions of different types, each able to recognize a particular antigen. When B cells become activated by T helper cells, they differentiate into plasma cells. These plasma cells become antibody-producing factories, flooding the bloodstream with antibodies that can bind to the antigen involved in this infection. Antibodies bind to the antigens on the surfaces of the pathogens, marking them for destruction by macrophages. Some of the B cells do not turn into antibody factories, but instead become memory B cells that may survive for several decades. Because of these memory B cells, the secondary immune response to a future infection by the same pathogen is swifter and stronger. This powerful secondary immune response is what gives immunity to some diseases after you have had them once or after you have been vaccinated.
So in this video, you saw a few different ways that this can take place. And so I'm going to run through a couple of things. First of all, an antigen-presenting cell presents the antigen to the T helper. So this is going to be an MHC2, which is going to activate a T helper with a CD4 marker on the cell. And once that happens, then it's going to stimulate cytotoxic T cells and B cells with that interleukin-2. And when it stimulates those B cells, the B cells then proliferate and differentiate. That's the T cell dependent method of activating B cells. And then the cytotoxic T cells will directly kill, but the B cells will make antibodies and indirectly kill. And so that's kind of what they're showing here. And then sometimes you have an infected cell, like a virus is in the cell, and then you have CD8 cells, which will react the cytotoxic T cell with an MHC1 in this phagocytic cell or infected cell. And so that is going to activate, and they poke them full of perforins and granzymes, and it causes them to lyse, basically. So that is the T cell, and then the B cell, you can see, proliferates and differentiates and makes these antibody-producing cells, which they show you how they neutralize the virus or toxins, and how they make memory cells as another thing. So that's a little bit about that. And now we're ready to look at the CD8 cells. So CD8 cells will respond to, again, some cell. Could be an antigen-presenting cell. It could be a virally infected cell. But the MHC1 presents the antigen to a CD8 cell. T helpers can also stimulate this with interleukin-2, as we saw in the video, and then, or the animation, and then it makes memory and activated CD8 cells. And the activated CD8 cells will produce perforins and granzymes to kill the infected cells. So it's a very specific killing. It only kills the cells when it's in this bond with another cell that is also presenting the same antigen in the MHC1. And so you have this very direct contact and then the killing by producing these cytokines, these perforins and granzymes, which blow up the cell. So that is the CD8. So we saw the CD4 and the CD8 Here is a little bit on the cytotoxic T cell, and I'm just going to show you again. They take up the virus, the cell takes up the virus, and in the process there's uncoating, and then it starts to make the virus parts. And when it makes those little virus parts, some of them get put into an MHC class 1, which is presented on the surface to a cytotoxic or CD8 cell, which then will secrete the perforins and the granzymes and other cytotoxins. And so perforins make it so that the granzymes can get in there and then they can die. They can kill off the cell. So perforins come first, then the granzymes. But they basically lyse the cell and then it goes away. So that is the cytotoxic T cell. Here is a lovely table which has all of the different subsets of the T cells and the type of marker that they have. Again, I'm really just focusing on the CD4 and CD8. There are others like the CD4L and the CD25 here for the regulatory, but you do need to know these functions. And so again, make sure you understand that the follicular helper helps drive B cell proliferation and the antibody class switching. 17 promotes the inflammation. Cytotoxic cell 2 can activate the macrophages also, but mainly we're going to focus on the fact that it contributes to allergies, type 1 allergies, extracellular antigens, including like protozoans and helminths. So protozoans, helminths, eukaryotic microbes, and allergies are what we're going to deal with the T helper 2. T helper 1 uses the macrophages, makes them angry macrophages, and makes this delayed hypersensitivity reaction, which happens days, hours, or days later. And also secretes interleukin 2 so that it activates the T cell mediated immunity. So just a review on that. And then regulatory prevents autoimmunity. That was the other thing. And prevents the microbiota from being damaged. It's not in this list here, but it does prevent our immune system from attacking the resident microbiota, and it also prevents the cells from going out of control, the immune system or the immune response from going out of control and making cells that are not necessary when the immune system has already cleared most of them. And then the cytotoxic T cell directly kills, and that's the only one we're talking about that's a CD8. Gamma delta T cells, they are going to react specifically and non-specifically, but they have a T cell receptor, but it's a different, like the little subunits that make up the T cell receptor are a gamma and delta polypeptide, where the other T cell receptors are alpha beta. So it's a slightly different shaped T cell receptor, and they can still react specifically, but they can also be rearranged to react to a lot of different non-specific antigens, and they're responsive to lipid antigens, which are typically not, something that we can respond to very well. So the B cell, again, can be activated specifically by or independent of the T cell, because it can do this process called capping. So it phagocytoses the microbe and then presents the antigen on the MHC2, or and when it presents it in the MHC2 or MHC1, it then activates the T helper cells or cytotoxic T cells, but they can also, depending on if it's an MHC1 or 2, but it can also, when it does the capping, it's all of these little antibodies are bound by the antigen, and they kind of cluster together into a group, and then they get endocytosed, and that is that's when it activates the B cell. So this is how it's presented the antigen and gets activated, and once it's activated, it will make the different B type cells. So here is the T cell dependent activation, and I'm just going to show you a little bit of how that goes. It's phagocytosed, it's combined with the phagosome-lysosome fuse, and then it gets combined with an MHC, in this case 2, which activates the B cell or the CD4 cells. which is a T helper cell. And then they proliferate. When it finds this gets activated, then it activates the B cells that are also presenting that antigen, and it proliferates and differentiates, makes many copies, and then the plasma cells are one of the differentiated type of cells, and they produce lots of antibodies. So that is one. This is so similar to some of the other animations. I didn't want to take time to really show the whole thing, but you can download the PowerPoint and watch it if you'd like to. This one, however, is the T cell independent method, and so I am going to show you this one.
The immune system can produce antibodies against millions of different antigens. B cells, which are constantly produced from lymphoid stem cells, are found in the blood and lymphoid tissues. Each B cell can synthesize only one of the millions of possible antibodies and displays this antibody on its surface. When an antigen meets a B cell having a surface antibody of the proper specificity, it complexes with the antibody. B cells that are not activated do not develop further. Stimulation of only the B cells that carry antibodies that react with antigen is referred to as clonal selection. Following antigen-antibody interaction, antigen-antibody complexes aggregate on the surface of the cell. This is called capping. The antigen-antibody complexes are then internalized. The activated B cell swells and begins to divide rapidly, producing a B cell clone. The B cells then differentiate into plasma cells and memory cells. Plasma cells produce the specific antibody that provoked its formation, whereas memory cells remain in circulation but do not produce antibodies. Memory cells can become activated by a later challenge by the same antigen.
So I just wanted to go through a few things in this animation before we go too much further. So this is the independent process. So B cells are made, they get their receptors, they get selected because the particular antibody matches the antigen matches the antibody. And that is called clonal selection. And so the others that don't get selected, they await activation themselves later. They don't develop further. But these, they do this process called capping, where the antigen-antibody complexes cluster together in one area, and then they are engulfed into the B cell. And the B cell then is activated. Once it's activated, it makes many copies of that clone, and then it differentiates into plasma cells and memory cells. And so once they're activated, the B cells will make regulatory cells which secrete interleukin 10 to regulate the T cell response. So it actually dampens the T cell response as well. And then the memory B cells will only replace B cells, and they remain in circulation to give us immunity to the disease. Plasma cells will create antibodies like these IgM antibodies right here, which then have indirect jobs of killing. They will increase phagocytosis or cause inflammation or increase, they sometimes will help the complement. The complement will poke holes in them. So that's the B cells. And the three questions that we have for this section 15.4 are: the blank cell activates cell-mediated immunity. And true or false, B cells can only be activated by T helper cells. And number 3, the CD8 cells are activated by antigen complex with which type of molecules? So the first answer here, the T helper 1 cell activates the cell-mediated immunity. And question 2, the B cells can only be activated by T helper cells. No, that's not true. They can be activated independent of T cells as well. And the CD8 cells are going to be activated by antigen complexed with MHC1. So 2418, the MHC1 contains the antigen that presents the antigen to the CD8 cells. That's how they're activated. So that was section 4.